Hi, my name is Lloyd Abraham, and today we will be talking about the role of platelets in renal injury. So now for a little background on the kidney. The kidney are bean-shaped organs that play an essential role in maintaining blood volume and pressure. Their main function is to regulate the balance of electrolytes in the blood, along with maintaining pH homeostasis. They also remove excess organic molecules from the blood, and it is by this action that their best known function is performed, which is the removal of waste products of metabolism. Now let's take an in-depth look at the kidney structure. Within each kidney, we have 18 lobes. Each lobe is called a renal pyramid. In each renal pyramid, we have multiple millions of nephrons. A nephron is a kidney functional unit that removes waste from the body. Each kidney has more than a million nephrons in its renal cortex, which gives it a granular appearance on sagittal cross sections. The nephron consists of renal corpuscles, a tubule, and a capillary network that originates from the small cortical arteries. Each renal corpuscle is composed of glomerulus, which is a network of capillaries, and a Bowman's capsule, which is the cup-shaped chamber that surrounds it. The glomerulus connects to a long convoluted renal tubule, which is divided into three functional groups. These consist of the loop of Henle, the proximal convoluted tubule, and the distal convoluted tubule. These then empty into the collecting ducts. The collecting ducts fuse together and enter the papillae of the renal medulla. The urine passes through the renal medulla as a fluid with high sodium content and leaves through the renal papillae, which then goes into the renal pelvis and into the bladder through the uterus. So acute kidney disease occurs when your kidneys suddenly become unable to filter waste products from your blood. Your kidneys lose their filtration ability, and dangerous levels of waste may accumulate, and your body's chemical makeup may get out of balance. So acute kidney disease develops rapidly over a few hours or even a few days. A result of acute kidney disease can be glomerulonephritis, which is characterized by the inflammation of either the glomeruli or the small blood vessels within the kidneys. But not all the diseases necessarily have a inflammatory component. A good example of this is IgA nephropathy. Um, Another another effect of acute kidney disease is glomerulosclerosis, which refers to the hardening of the glomerulus within the kidneys. It's a general term to describe scarring of the kidneys' tiny blood vessels and the glomeruli, um, which are the functional units within the kidney that filter the urine from the blood. So this scarring disturbs the filtering process of the kidneys and allows for protein to leak from the blood into the urine. The scarring of the glomeruli cannot be repaired, and many patients with glomerular sclerosis get worse over time until until their kidneys eventually fail. And this is what my paper looks at. Now let's look at my paper. The title of my paper was Platelets are relevant mediators of renal injury induced by primary endothelial lesions. It was written by Schwarzenberger et al. And was published by the American Journal of Physiology. Let's first look at the purpose of my paper. They had two purposes, which was to characterize the role of platelets in acute kidney disease, and they wanted to see whether the inhibition of the platelet activation would serve as a viable protective strategy for future diseases. Now let's look at the different treatment groups. Of the wild-type mice, we had three treatment groups, one of which was the platelet depletion group, which was seen through the use of antibodies. We then have the clopidogrel group, which was which uses receptors on the platelets to inactivate them. And then we have the control group. All mice were given a severe acute endothelial injury within their kidney in order to induce acute kidney disease. Now let's look at figure 1, where the researchers wanted to see whether the different treatment groups that we said were able to inhibit platelet activation um, actually worked. So first we have panel A, which is the control, and panel B, which is the platelet depleted group. And we're looking at a flow cytometry depicting different microparticles, which is MP, platelets, which is PL, and the immune complexes, which is IC. And we can see that in panel A that there is a dense amount of these components within the given areas. Well, when you compare that to panel B, you can see that the denseness is gone, that there's just aren't as many of those components within those areas as seen in panel A, which goes with our hypothesis that the um, treatment groups that are able to inhibit platelets actually showed a decreased amount of platelets within those groups. Now that the researchers know that the platelet inhibitors actually reduce the amount of platelets found, they wanted to investigate the effects of the platelet deletion on these endothelial injuries. So they took into effect the control group and the platelet depleted group, which was using the glycoprotein IBA antibody, and they wanted to see the different amounts of thrombin and specifically fibrin within the endothelial cells. So um, um, it's expected that if um, repair were to occur, we would have greater amounts of thrombin and fibrin found within the cells. However, and as a result, we can see that in C and D, in panel C and D, that the platelet depletion actually lowered significantly both the thrombi and fibrin percentages within those cells. And this results in um, panel B, where we can see that the peritubular um, capillary loss had actually decreased in the platelet depleted group than that of the control group. Now that they knew that the antibody dependent inhibitors of platelets worked in reducing the percentage of peritubular capacity, they wanted to look at whether the known clopidogrel, which was the receptor blocker for platelets, had the same effect, and if it even had a greater effect. So they first looked at clopidogrel's ability to decrease glomerular injury, which they saw that both in day 1 and day 3, compared to the placebo, it had a significant decrease in this injury. And then they looked at the capillary loss, where they found that only on day 3 they found a significant decrease in the amount of this capillary loss compared to the placebo. Finally, in panel C, they looked at the percentage of peritubular capillary loss, which they found that only on day 3 there was also a significant decrease found within the clopidogrel inhibitor. So from these figures, the researchers concluded that platelet depletion significantly reduced acute renal injury through the inhibition of glomerular injury, fibrin deposition, and TMA, which ultimately preserve the microvascular capillaries within the kidney. Finally, my discussion questions are: what are the first signs of an acute renal disease, and what other processes are involved with wound repair?